The slide presented includes the poster I created for my research project. Last summer, I investigated the regulation of oncogenic stress response in gastric cancer at the University of Miami in Dr. Alexander Zyka's laboratory. Before I discuss the major details of the project, I will be providing necessary background information on the topic at hand. Gastric cancer, also known as stomach cancer, is the buildup of abnormal cells located in the lining of the stomach. Gastric cancer is the fifth most common cancer diagnosis in the world. In the United States, approximately 27,000 individuals will be diagnosed, and unfortunately over 11,000 will die from this disease. Individuals are often diagnosed with late stages of gastric cancer because the symptoms associated are very common, including vague discomfort in the abdomen, nausea, heartburn, vomiting, and swelling. In addition, there are many risk factors affiliated with the development of gastric cancer. For example, an increased risk of stomach cancer is seen in people with diets that have large amount of smoked foods, salted fish, and pickled vegetables. These foods could be contaminated with certain bacteria such as H. pylori, which has been shown to be affiliated with gastric cancer. Helicobacter pylori is a gram-negative microaerophilic spiral bacteria discovered by Dr. Ray Marshall and Dr. Robin Warren in 1982. H. pylori is commonly transmitted through fecal contamination of food or water. H. pylori is more prevalent in locations where untreated water, crowded conditions, and poor hygiene are present. A fundamental response to tumorigenesis is enhanced cellular stress. Cellular stress can induce the production of free radicals, DNA damage, hypoxia, and nutrient deprivation. During these occurrences, p53, a protein that acts in tumor-suppressor, engages in activities such as cell cycle arrest, apoptosis, and DNA repair to prevent the development of malignancies. Previous studies conducted by my laboratory mentor, Dr. Zyka, observed that H. pylori suppresses oncogenic stress response, thus indirectly allowing tumorigenesis to occur. In order to accurately understand the specific metabolic pathways affected and proteins involved in gastric tumorigenesis, a model system was created. Through the cloning process, the TetOne-kRas vector grants a perfect opportunity to understand how oncogenic stress response functions. Previous research studies have reported that the mutated KRAS causes tumor initiation, which is a perfect reason as to why I was selected as our insert. The KRAS gene provides instructions for making a gene called KRAS, which is part of a signaling pathway that instructs cells to proliferate and differentiate. In order to prepare KRAS for ligation, PCR purification, restriction digest, gel, and gel purification were performed. The same steps were conducted for the TetOne vector. 8 individual colonies were selected for plasmid isolation. After isolation, gel electrophoresis was performed and analyzed to ensure that the insert and vector were present. From the 8 total samples, 2 were selected for sequencing in order to guarantee no mutations. The samples showed no mutations. With the creation of the model system, it will now be possible to easily detect which metabolic pathways are affected and what key proteins are involved in the regulation of oncogenic stress. Future studies will be conducted with this model system to understand gastric tumorigenesis and help identify potential targets in the prevention of gastric cancer. Overall, the main goal of this research is to prevent and cure one of the most lethal cancers in the world, gastric cancer. Thank you so much for allowing me to present.